Isolation streaking is a standard microbiology technique used to create individual colonies from a culture. Specific patterns used in isolation streaking may differ slightly, but the same basic process is followed to dilute the culture. We will demonstrate streaking in quadrants. The third or fourth streaks may not yield any colonies, but are done to ensure that at least one of the streaks provides single colonies. For isolation streaking, you need an inoculating loop, a bacterial culture, a plate for culturing, and either a Bunsen burner or an infrared bacteria sterilizer. Sterilize your inoculating loop using either an infrared sterilizer or a Bunsen burner. In this video, a Bunsen burner will be used. For further detail, see our video entitled Sterilizing an Inoculating Loop. The culture used to streak the plate may be in liquid broth or on an auger slant or plate. In this video, a slant is used. Without touching the loop to any surface, carefully open the lid or cap of the bacterial culture. If the culture is in a tube, pass the mouth of the uncapped tube through the flame. Cool the loop by inserting it into the auger of the slant or plate in an area without bacteria. Use the loop to pick up a very small amount of the bacterial culture. This small quantity contains all the bacteria needed for streaking a plate. Again, sterilize the opening of the culture tube and replace the lid. Try not to touch the sides of the culture tube when inserting or removing the loop. As you work, lift the lid of the plate only enough to allow streaking. Holding the lid over the plate helps protect it from airborne contaminants. Imagine that the plate is divided evenly into 4 regions. Beginning at the outer edge of the plate, lightly rub the loop in a zigzag pattern across one quadrant. To avoid tearing the auger, be sure to streak lightly and not too quickly. Once the first zigzag is complete, replace the lid on the plate and sterilize the loop. Because the sample used to make the first streak came directly from the culture, the growth of the bacteria in the first streak will be denser than in the other streaks. Rotate the plate a quarter turn, open the lid, cool the loop by sticking it into the auger at an uninoculated edge of the plate. Open the lid and again create a zigzag pattern with the inoculating loop, this time in the second quadrant. Remember that the loop is again sterile and that in order to collect and spread some bacteria, you must run the loop through some of the zigzag in the first quadrant. Make sure that only the first swipe of the second zigzag crosses the first streak. Again, sterilize the loop, rotate the plate, and cool the loop. Create a zigzag streak in the third quadrant, this time allowing the first swipe of the zigzag to cross the second streak. A final time, sterilize the loop. Rotate the plate, cool the loop, and streak. Streak the fourth zigzag in the fourth quadrant, allowing the first swipe of the fourth zigzag to cross the third streak. Isolation streaking serves to dilute bacteria to ensure that somewhere on the plate there are individual colonies. After incubation, the region streaked first will have the greatest density of colonies, and the region streaked last will have the lowest density.